In Chapter 18, we're going to talk about the specific immune response. And we're just going to do an overview in this first lecture. We've seen this slide before, so this is really important to know for the next exam. Make sure you can tell me the difference between the two branches of the immune system. We have the non-specific immune response, also called innate immunity. And then we have the specific immune response, which is also called adaptive or acquired immunity. So the last chapter, we focused on the non-specific immune response. So remember, it's non-specific. It goes after everything. It's not picky. It's going to react fast and with the same speed and strength each time. But it doesn't remember that it's seen a particular pathogen before. And it includes things like physical barriers, complement system, etc. We're going to be looking at the specific immune response in this chapter. And it's called specific because the B and T lymphocytes are very specific for what they recognize. So you've got millions and millions of different B and T lymphocytes, and each one recognizes only one specific pathogen. So if a B lymphocyte comes up and looks at a measles virus and it's not its match, it ignores it. So you have to find the right B and T lymphocyte that recognizes the measles virus in order to respond to that. The first time you're exposed to a pathogen, the B and T lymphocytes are going to respond slowly, but they're going to create some memory cells. And the next time you're exposed, they're going to react really fast and strongly, and so you don't get sick the second time. So the specific immune response has to kind of learn. So that first response is slow, you do get sick, but the second time you learn that, oh, that was awful, now I've got these memory cells, and now I'm going to react really fast and strongly and not get sick. So that's why it's also called the adaptive or acquired immunity, because you have to learn, you get that slow response at first and then the fast second response. So there's three major functions of the specific immune response. So it has to recognize non-self, things that don't belong in your body. And then it has to respond to them. So when the B and T lymphocytes find their match, they're going to start dividing and they're going to produce these effector cells. And the effector cells are the ones that are going to go and do battle and will get rid of the pathogen or foreign material, whatever is not supposed to be there. And we're also going to be creating a second set of cells called memory cells. So they're there for the second time you're exposed to the same pathogen. The memory cells remember, and they're going to react really fast and strong the second time, and then you don't get sick the second time. There's two branches of the specific immune response. So to start this is really important to know. There's humoral immunity and cell-mediated immunity. So humoral immunity involves the B lymphocytes. And when the B lymphocyte finds its match, it starts dividing, and the effector cells that it produces to do battle are called plasma cells. And these plasma cells produce antibodies. And antibodies are big proteins that are going to bind to the specific pathogen that's outside the cells that are in the body fluids. And the antibodies are like big red flags, so once you tag something with an antibody, the other white blood cells will come over and get rid of it. So the reason that is called humoral immunity is that humors is a very old term that means body fluids. So that's why it got that name. Cell-mediated immunity is going to involve the T lymphocytes, and specifically the cytotoxic T lymphocytes. And the cytotoxic T lymphocytes are going to go around and find any infected cells, cancer cells, or transplanted cells and kill them. So for example, if you have any type of pathogen that's inside of a cell, the antibodies are not going to be able to get those because the antibodies are big proteins. They're only going to be out in the body fluid. So we needed to have something that could find the infected cells, things that were inside the cell, and that's where cell-mediated immunity comes in with these cytotoxic T cells. So here's just a nice picture that I drew for the specific immune response and the two branches. So we've got humoral immunity, so that involves your B lymphocytes producing antibodies to a specific pathogen, and that will bind to the pathogen, maybe this virus here, out in the body fluids. But if you have the virus inside the cell, the antibodies can't get to that. So that's where cell-mediated immunity comes in. So these cytotoxic T lymphocytes will find these infected cells, or maybe it's a cancer cell or transplanted cell, and will destroy those cells. So there's 4 different characteristics of the specific immune response. You don't need to memorize this list, but just understand what's going on. So the first one is self-tolerance or immune tolerance. So we don't want our B and T lymphocytes attacking our body parts. So that would be really bad, and that is what happens in an autoimmune disease. So when we're making the B and T lymphocytes in the red bone marrow, it's just random what kind of receptors they end up with and what they recognize. So some of them do recognize our body parts. So when they're maturing in the bone marrow or the thymus, we're going to check, and if any of those B and T lymphocytes react to body parts, they will either be killed or they're going to be inactivated. And when they're inactivated, we just call them anergic, because an means without and erg means to work. So it just means they're not working. So if there's a failure of that, we can end up with the autoimmune disease. The next characteristic of the specific immune response is diversity. So we want to be ready for anything because we never know what kind of pathogens we're going to be exposed to during life. So when our B and T lymphocytes are produced, there's millions and millions of different receptors that they could end up with. So each one's going to be very specific for which pathogen they recognize, and that way we're ready for just about anything. Another characteristic is specificity. So each B and T lymphocyte has an antigen receptor that's only going to recognize one specific pathogen. And if it's not its match, it will not react to the pathogen. So here's just an example of the receptors. So the B lymphocytes actually use an antibody as their receptor. So it'll be very specific to which pathogen it binds to. The T cell receptors look just a little bit different, but again, they're very specific for what they're going to recognize too. And then the last characteristic of the specific immune response is memory. So the first time you're exposed to a pathogen, so let's say you got exposed to the chickenpox virus, you're going to have the B and T lymphocytes that recognize the chickenpox virus. They will get stimulated to divide, and they're going to produce 2 different types of cells. So the effector cells are going to go do battle with the virus, and then the memory cells will not participate in the battle. They'll just kind of stand on the sidelines and root for the home team. And this primary response, the first time you're exposed to the chickenpox virus, is slow. It's not as strong, and so you will get sick with the chickenpox. But if you're exposed to the chickenpox virus again years later, you have those memory cells that are still with you, and oftentimes they can last your whole life. And these memory cells will react much faster and stronger the second time around, and so you don't get sick again with the chickenpox. So this is just showing you a couple of different B lymphocytes here. And let's say maybe this one in the middle is the one that recognizes the chickenpox virus. So it's going to get stimulated to divide, and it's going to form 2 sets of cells. So the effector cells are the ones that are going to go and do battle with the virus. So in the B lymphocytes, the effector cells are called plasma cells, and they're going to secrete lots of antibodies to bind to any of the chickenpox virus that are outside the cells. And then the second group of cells we're going to produce are memory B cells. So memory B cells will not participate in the battle, but they're there for the next time you're exposed, and they're going to react much stronger and faster so you don't get sick a second time. So start this graph. This is really important to understand. This will definitely be on the test. So the primary immune response, so the first time you're exposed to chickenpox, notice that it takes about 10 days before you start to produce antibodies. And in those 10 days, the chickenpox virus has been having a party in your body and making you miserable and causing all kinds of sores everywhere. But then you start producing antibodies, you start fighting off the virus, and then you get better. So the second time you're exposed to the chickenpox virus, the secondary response, notice that it only takes you about 3 days to start producing antibodies. So these memory cells that you produced on the primary response, they're sitting there waiting, and if they see the chickenpox virus again, they remember how bad that was, and they're like, we're not doing that again. So those memory cells are going to react really fast and produce a much stronger response as well. So notice how you produce a lot more antibodies on the secondary response. So that's why you don't get sick with the chickenpox virus if you're exposed again.